I'm Randall Buck.
I'm Ryan Fosnot.
I'm Jade Houston.
And I'm Truman Rowland.
Our bodies have evolved biological systems to combat threats, internal and external. Viruses, bacteria, and cancer. Adaptive immunity is our final line of defense against these monsters. A smart weapon, immune cells learn the biochemical signature of our adversaries to win the battle. However, our current therapies, such as chemotherapy, are dumb, simply put. Critical tissues become collateral damage. Therapies for metabolic disease, brought on by socioeconomic plights, are riddled with insufficiencies and side effects. Looking to our immune system, we propose using gene editing and microfluidics to create proteins that don't just fight disease, but preserve health. With modern DNA technology, we make possible the creation of protein arsenals that target cancer cells alone and fight metabolic diseases without such crippling off-target effects.
To address this, in collaboration with Moffitt Cancer Research Center, we have developed Microfab Fluidics, a novel lab-on-a-chip system which consists of a 5-layer design to accomplish cell pair encapsulation while remaining leak-free and sterile.
Utilizing SLA resin printing, we have forged a chip that can expedite new drug screenings from what would take centuries down to mere months. Our device intakes up to 4 reagents, and for this specific use case, microscale volumes of cells infected with randomized protein genes. Within the lab-on-a-chip, the infected cells are mixed with cells that have drug targets on the cell surface, and which glow green if activated. These cells are co-encapsulated into water droplets suspended in oil. The droplets are encased in a protective hydrogel layer and incubated to allow the cells to glow green. The output of the lab-on-a-chip is sorted with a flow cytometer, keeping our glowing green droplets containing a new candidate protein drug.
We have calcium ions in our cell suspension that will then cross-link with alginate and DMAP. To prepare, we begin running our pump, which begins to output fluid. Once we see droplets begin to form, we connect the cap, which has a hose barb that points directly down and is connected with a lure lock on top. In about 40 seconds, we filled 10 milliliters of oil. After collecting droplets, we invert the solution to mix the cross-linking alginate with our droplets until we get a suspension of oil droplets and hydrogel droplets.
Microfluidics is the gateway not only to revolutionary specificities and efficiencies in targeted cancer immunotherapies and metabolic disorder treatments, but making this technology a reality for labs of all kinds.